RNA editing. RNA editing is post-transcriptional processes that alter the information encoded in gene transcripts. Nowadays, such changes have been observed in rRNA, tRNA and mRNA molecules of eukaryotes, but not prokaryotes. RNA editing has been reported in protozoa and viral RNAs, mitochondrial, nuclear, chloroplast RNAs. RNA editing processes show great molecular diversity, and this diversity of RNA editing mechanisms includes nucleoside modifications such as adenosine to inosine and cytidine to uridine deaminations, as well as non-templated nucleotide additions and insertions. The RNA editing processes alter the information content of gene transcripts by the following methods. 1. Editing by base modification, that is deaminase, and 2. Editing by insertion or deletion of nucleotides. Editing by base modification, that is deamination. Deamination includes two types of modifications such as cytidine to uridine and adenosine to inosine deaminations, as well as non-templated nucleotide additions and insertions. Cytidine to uridine or C to U editing. The first type of RNA editing is C to U editing, which results in the substitution of one base for another base. This type of RNA editing was discovered in studies of apolipoprotein B, that is ApoB genes, and in mRNA of rabbits and humans. Apolipoproteins are blood proteins that transport certain types of fat molecules in the circulatory system. ApoB contains 29 exons separated by 28 introns. In the liver, the ApoB mRNA encodes a large protein consisting of 4563 amino acids long. The gene is expressed in cells of both the intestine and liver. In intestine and liver, transcription produces a pre-mRNA that must be spliced to produce the mRNA and translated into protein. In the intestine, the ApoB mRNA directs the synthesis of a protein which is only 2153 amino acids long. This editing involves cytidine deaminase that deaminates a cytidine base into a uridine base, generating a translation termination codon UAA, which results in the truncated apolipoprotein. UAA is one of the three codons that terminate polypeptide chains during translation. If a UAA codon is produced within the coding region of an mRNA, it will prematurely terminate the polypeptide during translation, yielding an incomplete gene product. In the intestine, translation of the mRNA stops at codon 2153, forming apolipoprotein B48, a protein that contains 2152 amino acids and that aids in the absorption of dietary lipids from the contents of the intestine. In liver, the RNA editing process occurs normally producing apolipoprotein B100, a protein which contains 4563 amino acids that is essential for the transport of cholesterol and other lipids in the blood. The C to U conversion is catalyzed by a sequence specific RNA binding protein with an activity that removes amino groups from cytosine residues. Another example of RNA editing has been documented for an mRNA specifying a protein, the glutamate receptor present in rat brain cells. C to U editing often occurs in the mRNA of flowering plants. Different plants have different degrees of C to U editing. For example, 8 editing events occur in mitochondria of Funaria hygrometrica, a moss, whereas over 1700 editing events occur in Cetus engelmanni, that is Engelmann's quillwort. C to U editing is performed by members of the PPR family, that is pentatricopeptide repeat protein family. Angiosperms have a large number of PPR families acting as trans factors for cis elements lacking a consensus sequence. Arabidopsis thaliana has around 450 members in its PPR family. There have been a number of discoveries of PPR protein families in both chloroplast and mitochondria. More extensive mRNA editing of the C to U type occurs in the mitochondria of plants, where most of the gene transcripts are edited to some degree. Mitochondria have their own genomes and protein synthesizing machinery. In some transcripts present in plant mitochondria, most of the cytidines are converted to uridine residues. Adenosine to inosine editing, AI editing. This is the main form of RNA editing in mammals and occurs in regions of double-stranded RNA. The RNA editing enzymes, adenosine deaminases acting on RNA, is involved in the hydrolytic deamination of A to I editing, that is adenosine to inosine. A to I editing can be specific, meaning that a single adenosine is edited within the stretch of double-stranded RNA, or A to I editing can be promiscuous, which means 50% of the adenosines are edited. Specific RNA editing events occur within short duplexes. Example, those formed in an mRNA molecule were intronic sequence base pairs with a complementary exonic sequence, while promiscuous RNA editing occurs within longer regions of duplex. Example, pre or primary miRNAs, duplexes arising from transgene or viral expression, duplexes arising from paired repetitive elements. These effects include altered siRNA or miRNA target populations, heterochromatin formation, alteration of coding capacity, cytoplasmic sequestration, nuclear sequestration, endonucleolytic cleavage by Tudor SN, inhibition of siRNA and miRNA processing and altered splicing. This type of RNA editing occurs in humans, for instance ALS, that is amyotrophic lateral sclerosis, which is the most common adult onset motor neuron disease, express unedited glutamate receptor 2 mRNA at the QR site in a disease specific and motor neuron selective manner. Glutamate receptor 2 is a subunit of the AMPA receptor, that is alpha amino 3 hydroxy 5 methyl 4 isosol propionic acid, and it has a regulatory role in the calcium permeability of the AMPA receptor after the genomic Q codon is replaced with the R codon in mRNA by adenosine to inosine conversion, which is mediated by adenosine deaminase acting on RNA 2, that is ADR2. Therefore, ADAR2 activity may not be sufficient to edit all glutamate receptor 2 mRNA expressed in the motor neurons of ALS patients. RNA editing at the glutamate receptor 2 QR site is catalyzed by adenosine deaminase acting on RNA 2, a member of the ADR family. ADR2 specifically catalyzes the adenosine to inosine editing at the glutamate receptor 2 QR site, and two isoforms ADR2A and ADR2B are expressed in human brains and spinal cords. ADAR2 specifically catalyzes the adenosine to inosine editing at the glutamate receptor 2 QR site, and two isoforms ADAR2A and ADAR2B are expressed in human brains and spinal cords. ADAR2 catalyzes adenosine to inosine editing in the double stranded RNA structure in both coding and non-coding regions of various transcripts expressed in mammalian brains. When this type of RNA editing occurs in the reading frame, the resulting changes in the amino acid residues alter the biological function of the translated molecules, including 5-HT2C receptors, glutamate receptors, and glucokinase receptors. Adenosine to inosine RNA editing at the glutamate receptor 2 QR site is specifically catalyzed by ADAR2. ADARs act on double stranded RNA, which is formed with the distal end of exon 11 that includes the coding sequence for the QR site and the exon complementary sequence in intron 11 of glutamate receptor 2 pre-mRNA. Editing by insertion or deletion. This is the complex type of RNA editing which occurs in the mitochondria of trypanosoma, a group of flagellated protozoa that cause sleeping sickness in humans. In this case, uridine monophosphate residues are inserted into gene transcripts, causing major changes in the polypeptides specified by the mRNA molecules. The trypanosome mitochondrial type of editing is illustrated in this figure, which shows the sequence of the DNA coding strand, the pre-edited RNA transcript and the edited mRNA of the cytochrome B gene of Leishmania tarantole. Cytochrome B is a protein involved in the production of chemical energy. This RNA editing process is mediated by guide RNAs which transcribe from distinct mitochondrial genes. The guide RNAs contain sequences that are partially complementary to the pre-mRNAs to be edited. Pairing between the guide RNAs and pre-mRNAs results in gaps with unpaired adenine residues in the guide RNAs. The guide RNAs serve as templates for editing, as uridine monophosphate residues are inserted in the gaps in pre-mRNA molecules opposite the adenines in the guide RNAs. Alternative mRNA editing. Alternative uridine to cytidine mRNA editing was first reported in Wilms tumor 1 transcripts, and non-classic G to A mRNA changes were first observed in heterogeneous nuclear ribonucleoprotein K transcripts in both normal and malignant colorectal samples. The latter changes were also later seen in alongside non-classic U to C alterations in brain cell tryptophan hydrolase 2 transcripts. Although the reverse amination might be the simplest explanation for U to C changes, transamination and transglycosylation mechanisms have been proposed for planned U to C editing events in mitochondrial transcripts. A recent study reported novel G to A mRNA changes in WT1 transcripts at 2 hotspots, proposing the apolipoprotein B mRNA editing enzyme catalytic polypeptide 3A as the enzyme implicated in this class of alternative mRNA editing. RNA editing in plant mitochondria and plastids. This is the only type of RNA editing seen in the plant's mitochondria and plastids, and this type of conversion, that is C to U and U to C, is very rare. The sites for RNA editing are found mainly in the introns, coding regions of mRNA and other non-translated regions. In fact, RNA editing can restore the functionality of tRNA molecules. The RNA editing sites are found primarily upstream of mitochondrial or plastid RNAs. The specific positions for C to U RNA editing events have been well studied in both the mitochondrion and plastid. Members of the expansive PPR protein family and specific members of the multiple organellar RNA editing factor, that is MORF family, have been shown to function as transacting factors for RNA sequence recognition. As some of these MORF proteins have been shown to interact with members of the PPR family, it is possible that MORF proteins are components of the editosome complex, a multi-protein complex that catalyzes the editing of RNA. An enzyme responsible for the trans or deamination of the RNA transcript in this editing remains elusive, though it has been proposed that the PPR proteins may serve this function as well. RNA editing is very essential for the normal functioning of the plant's translation and respiration activity. Editing can restore the essential base pairing sequences of tRNAs, restoring functionality. It has also been linked to the production of RNA edited proteins that are incorporated into the polypeptide complexes of the respiration pathway. So, the polypeptides synthesized from the unedited RNAs would not function properly and hinder the activity of both mitochondria and plastids. C2U RNA editing can create start and stop codons, but it cannot destroy existing start and stop codons. A cryptic start codon is created when the codon ACG is edited to AUG. RNA editing in viruses. RNA editing in viruses such as measles, mumps or parainfluenza are used for stability and generation of different proteins. Viral RNAs are transcribed by a virus encoded RNA dependent RNA polymerase, which is prone to pausing and stuttering at certain nucleotide combinations. In addition, up to several hundreds of non-templated adenosines are added by the polymerase at the 3' end of nascent mRNA. This shifts the reading frame, which generates a different protein. These adenosines help stabilize the mRNA. Furthermore, the pausing and stuttering of the enzyme RNA polymerase allows the incorporation of 1 or 2 guanosines or adenosines upstream of the translational codon.